The microneutralisation assay, or MNA for short, is a live virus cell-based assay. It is used to determine antibody concentrations to give an indication of how good a vaccine may be. For example, testing antibody response against SARS-CoV-2 during the COVID-19 pandemic. It is used extensively at UKHSA in collaboration with vaccine developers to predict how well vaccines protect against and reduce the severity of infection with SARS-CoV-2. Vaccines are tested and samples collected as part of clinical trials. Following vaccination, participants donate blood samples. Serum, the fluid part of blood, is then isolated from these blood samples in the lab. This serum contains a person's antibodies, which are a major part of an immune system. The microneutralisation assay starts by mixing this serum, containing antibodies, with a predetermined amount of virus, for example SARS-CoV-2. During an incubation period, any antibodies in the serum sample that recognise the virus will bind to it and prevent it from being able to infect. This is referred to as neutralization. An adherent cell line, VDEC, is commonly cultured for use in this assay to test for levels of infection in vitro. Other cell lines can be used, but VDEC cells have been found to be susceptible and permissible to SARS-CoV-2 infection and replication. When cultured in the lab, these cells form a single layer, or monolayer of cells, on the base of a flask or plate. This assay is predominantly carried out on 96 well plates, and cells are seeded the day before they are needed, aiming for around 90% confluency, or plate surface coverage, on the day of the assay. In the MNA, the serum and virus mixture is added to the wells containing cell monolayers. This is then incubated to allow adsorption or infection of the virus. In the absence of specific antibodies, viruses can infect a cell by locking onto a receptor and entering it. Cells infected with SARS-CoV-2 express the viral spike protein on their surface. The virus then uses the cell's own machinery to replicate itself, ultimately being released from the host cell and infecting more cells. Virus particles that have been bound by antibodies may be neutralized. These neutralized virus particles are not able to enter or infect cells, remaining in the liquid in the well. As it would be difficult to visualize a single infected cell, the monolayer is left to incubate for roughly 24 hours in a viscous media to control the spread of replicated virus. This results in small areas, commonly referred to as foci, of infection. These foci are then large enough to be visualized using a process called immunostaining. Immunostaining uses antibody binding to visualize where cells have been infected. First, cells are fixed. This process preserves the cells in the state that they are in and removes the remaining liquid. This also stops any more virus replication or infection. Cells that have been infected will have the viral antigen on their surface. A primary antibody that recognizes this antigen is added to bind to these infected cells. Next, a secondary antibody is added that will bind to the first antibody. The secondary antibody is attached to HRP, or horseradish peroxidase. With the addition of a substrate, for example true blue, this HRP can be visualized, reacting with the substrate and turning blue. The final product is a monolayer of fixed cells with blue spots that are visible to the naked eye. By counting these blue spots, neutralizing antibodies present in the original patient sample can be determined. In short, fewer foci equals more antibodies, and vice versa. This information is then sent back to the vaccine developers and forms part of the data package needed to support vaccine licensure and use.